Hello, this is Dr. S. Padma from the Department of Biochemistry and Nutrition, Bhavan's Vivekananda College of Science, Humanities and Commerce, Sainipuri, Secunderabad. Here, I am discussing the research article titled Spermidine Attenuates Bleomycin-Induced Lung Fibrosis by Inducing Autophagy and Endoplasmic Reticulum Stress-Induced Cell Death in Mice. This research article was published in the journal Experimental Molecular Medicine by the authors Sung Woo Park, Baek Er, and Hong J. Coming to a brief background of the corresponding author, the author research mainly focuses on the therapeutic strategies for idiopathic pulmonary fibrosis. Several important publications by this author on idiopathic pulmonary fibrosis and other conditions related to lung pathology are given below. Few of the articles have been published in journals like Journal of Immunology, Science Reports, and so on. Moving on, now I will give you a brief introduction about what is this idiopathic pulmonary fibrosis. This is a progressive, devastating disease of the lungs and is characterized by injury to the alveolar epithelial cells and subsequent proliferation of activated fibroblasts known as myofibroblasts. When these myofibroblasts accumulate, this leads to the excessive deposition of the extracellular matrix, resulting in irreversible distortion of the lung parenchymal structure. Here, I am showing you a brief picture description of the progression of this into idiopathic pulmonary fibrosis. As you can see here, whenever the epithelial cells are subjected to the injury, there is a release of the inflammatory mediators and migration of the inflammatory cells like the macrophages, T cells, and the neutrophils. These are causing the conversion of these fibroblasts to myofibroblasts, and these myofibroblasts are converted to the extracellular matrix deposition in the lungs, leading to the condition that is idiopathic pulmonary fibrosis. Extensive research has gone into identifying the therapeutic strategies for this particular condition. Several experimental studies have been carried out on animal models as well as by using the interventional approaches, that is the study of effect of gene therapy on established fibrosis. Similarly, delivery vectors have also been used as therapeutic strategies, and pulmonary administration of different drugs has been also explored to achieve site-specific delivery. Today, we are going to look at one of such therapeutic approaches, including the mouse models, and explaining the strategies as well as some of the therapeutic interventions which could be possible for the treatment of this idiopathic pulmonary fibrosis. Moving on, idiopathic pulmonary fibrosis is initiated by repeated injury as well as shortened survival of alveolar epithelial cells. Lung fibrosis occurs due to repair processes which are abnormal as a result of the increasing age of the affected individual and is associated with telomerase attrition and senescence. Among the several biomolecules that can be used for therapeutic strategies, polyamines have been demonstrated for their antioxidant and anti-inflammatory effects in animal models. One such polyamine, spermidine, is ubiquitously distributed in living organisms and is involved in various biological processes. Spermidine is well known for its beneficial effects in the treatment of chronic inflammatory diseases. Spermidine levels have been known to be reported to be decreased in idiopathic pulmonary fibrosis compared to healthy controls. So, this present study is carried out to evaluate the therapeutic effect of spermidine on the mouse model for idiopathic pulmonary fibrosis. Coming to the methodology which has been adopted in this article, one set of experiments was establishing the idiopathic pulmonary fibrosis cultures from the patients, and another one is setting up the mouse model. So for this, setting up the mouse model for idiopathic pulmonary fibrosis, these cells, embryonic cells, were isolated, cultured, and the suspension was made, and this pulmonary suspension was used for magnetic purification of the uh, pulmonary cells, and the isolated cells were then maintained in 10% FBS in 25 millimolar herpes pH 7. Likewise, the fibroblasts from the lung biopsies of normals as well as IPF patients were collected, isolated by mechanical dispersal by trypsin digestion, and these cells were again isolated and maintained in 10% FBS in 25 millimolar herpes. Further to these experimental strategies, several parameters were conducted in order to validate the use of spermidine as a therapeutic target for the treatment of idiopathic pulmonary fibrosis. Some of the assays which were carried out include the apoptosis assay, galactosidos immunofluorescence staining, histological assays, histochemical staining, trichome staining, hydroxyproline levels in lung, pro-inflammatory and pro-fibrotic cytokine levels, as well as subjecting the data to statistical analysis. Now let us see the results of each of these experiments. The first study was to look at the anti-apoptotic effects of spermidine on BLM, that is bleomycin induced alveolar epithelial cells. Here, this graph shows you the flow cytometric analysis of the cells which are treated with bleomycin and which are treated with spermidine. So this is a controlled population where only bleomycin and spermidine have not been added, and this is a test population where only bleomycin is added. There you can see that there is lot of apoptosis happening in cells. When bleomycin and spermidine were added together, this is comparatively reduced further to complete exclusion of bleomycin where apoptosis was very much minimal. So this has been further confirmed by the percentage of necrosis and percentage of apoptosis happening in these different conditions. Histopathological examination of these samples, that is the control as well as the bleomycin treated cells, they showed increased S-gal activity after exposure and treatment with 100 micromolar concentration of spermidine as seen in this below figure reverse the senescence in these cells. The cell extracts were also analyzed by immunoblotting for the expression of these and apoptotic proteins P61 and P21 in whole lungs from mice. As it is seen here, this P16, which is a tumor suppressor gene, and P21, which are tumor suppressor genes, the expression of this P16 was found to be elevated in the cells which are treated with bleomycin, indicating that this is stimulating the death of cells, whereas in the cells which are treated with bleomycin and spermidine, the concentration of this P16 and P21 was substantially reduced, and furthermore, when only bleomycin was eliminated and only spermidine was included in the culture. The next set of results show the fold change in the concentration of the P16 as well as the P21 proteins in the presence and absence of bleomycin and spermidine. Here, you can see that in the presence of bleomycin, the levels of percentage change or expression of the protein is very much less as compared to this condition. Between P61 and P21, P21 showed a remarkable recovery when added with spermidine and bleomycin. As I told earlier, subsequent experiment included testing the levels of the pro-inflammatory mediators and prokaryotic cytokines in the mouse lungs. The pro-inflammatory mediators which were tested and pro-fibrotic cytokines which were tested included the interleukin-1, TNF-alpha, and the T-transforming growth factor beta. Here, again, the conditions are in such a way that these are the untreated cells. These are treated only with the bleomycin, and these are treated with bleomycin as well as spermidine. So, in case of interleukin-1, there is an increased concentration of this when only bleomycin was used, indicating that this bleomycin is causing the increase in the pro-inflammatory mediators. But when spermidine was included in the reaction, the levels of this interleukins increased to a certain extent compared to other mediators. So both IL-1, TNF-alpha, and the TGF-beta, all of them have also shown an increase in the levels of these anti-pro-inflammatory mediators and pro-fibrotic cytokines in the presence of spermidine and bleomycin. Coming to the next set of analysis, this included analyzing the total cell count, the macrophage count, neutrophils, and the lymphocytes, and indicating the role of spermidine, which is going to prevent the bleomycin-induced lung inflammation and fibrosis development. Further to this, hydroxyproline levels, which are an index of the formation of the collagen, which is part of the extracellular matrix, was also found to be very high in bleomycin treated culture as compared to the culture which was treated with the combination of bleomycin and spermidine. Here, subsequently, spermidine was also added externally to see the increases in the pro-fibrotic and pro-inflammatory mediators levels, which are increased by bleomycin. So here we have seen that this treatment, exogenous treatment of cells with this spermidine decreases the expression of some of these reactive substances related proteins, as can be seen both in the SDS page as well as in the bar graph analysis. And this shows the capsase 3, which is an important protein that is involved in the apoptotic mechanism, that is highly elevated in the case of bleomycin treated cells, indicating that bleomycin pushes the cells more into apoptosis, whereas in case of spermidine, these levels are not elevated, but these are controlled, so showing that it is an anti-apoptotic effect on the growing cells. To conclude, some of the key conclusions of this study were the treatment with spermidine is in induced bleomycin induced cells increases in the capillary cell death, which is composed of the main pathophysiology of idiopathic pulmonary fibrosis. Exogenous spermidine suppresses the bleomycin induced increase in cellular senescence. Accumulation of excessive spermidine may be harmful both in vitro and in vivo. Decrease in caspase-3 protein expression and the number of tunnel-positive cells suggests that spermidine inhibits BLM-induced apoptosis in the lung. And also, finally, it is also important to conclude from this data that exogenous spermidine administration attenuated lung inflammation and fibrosis in established BLM-induced experimental fibrosis. Coming to the key findings of this paper, this paper is the first paper to report the effect of the exogenous spermidine on experimental lung fibrosis and to investigate its possible mechanism. This paper further concludes that spermidine may be a promising therapeutic agent for fibrotic lung diseases, including IPF. So, this article assumes significance as it demonstrates the use of spermidine as a therapeutic agent in the treatment of idiopathic pulmonary fibrosis. Further, the workflow and the experimental strategies are well designed and can be adapted to demonstrate the effectiveness of treatment using polyamines. Thank you.